We're now on part 3 of aerobic respiration, and we're still in the matrix of the mitochondrion, and this next stage is called the Krebs cycle. In the previous stage, the link reaction, we produced acetyl coenzyme A, and that's where our story ended. So we're going to pick it up at this point here. So we've got our acetyl coenzyme A. Now what happens to the acetyl coenzyme A? It's going to be split back into acetate and coenzyme A. That coenzyme A is going to go back to the link reaction, but it's the acetate that we are interested in. Now, what's going to happen to the acetate? It's a 2-carbon structure, 2-carbon compound, and that acetate is going to combine with this 4-carbon compound called oxaloacetate. And then when the oxaloacetate and the acetate combine, it obviously makes a 6-carbon compound called citrate. Citrate then loses a carbon dioxide and it loses a hydrogen. Now, when it loses a carbon dioxide, we say that's called decarboxylation. And when it loses a hydrogen, we say that's called dehydrogenation. So as a result of dehydrogenation and decarboxylation, our 6-carbon compound has now turned into a 5-carbon compound. At this level, you don't need to know the name of that 5-carbon compound. That 5-carbon compound will undergo another series of decarboxylation and dehydrogenation, and that turns into a 4-carbon compound. Incidentally, it's a coenzyme NAD here and also there that's going to be picking up those hydrogens, and therefore they're going to become reduced NAD. You'll have a look in the next stage, oxidative phosphorylation, what happens to those reduced NADs. This 4-carbon compound is going to then go on and lose a phosphate, and that phosphate is going to be picked up by a molecule of ADP, therefore turning into ATP, and you already know that that's what we call substrate level phosphorylation. This 4-carbon compound is then going to be dehydrogenized and is going to be picked up by a coenzyme called FAD. Now, this is the first time you've been introduced to this coenzyme, but it does the same job. It carries the hydrogen from the Krebs cycle to the next phase, which is oxidative phosphorylation. So therefore that FAD is going to become reduced FAD. And then this 4-carbon compound is going to be dehydrogenized one step further, and then obviously that'll be picked up by the coenzyme NAD and taken to the next stage. All of these steps ends up with you having the original 4-carbon structures you started off with, which is oxaloacetate. And then that oxaloacetate can obviously combine with another acetate as and when it arrives. And that is the Krebs cycle.